we'll get to know each other a little bit more. And so CCMB has been around for, I just confirmed with Soren, 20 years. And it was really a response to the Human Genome Project, which at the time we began to recognize that we needed to develop computational methods, and we needed computational biologists to, analyze genomic data. And since then, CCMB has gained that strength, and since then it has evolved to also analyze other types of omics data sets. And so, and I know that the RNA Center is now heavily involved in a big project called the Human RNA Project, which is an international initiative to create a comprehensive reference map of all RNA molecules and their chemical modifications. And so one of the goals of this symposium is to learn about each other and to identify exciting research opportunities for collaboration. And so the plan for today is to have some talks in the morning, and then between coffee breaks, or talks and then coffee breaks, and then a breakout session where we'll split into 4 different groups where we can have small discussions. And I'll put up a slide with what are the discussion themes before we start. And then we'll have a lunch and a panel discussion. And the title of our panel discussion is "How can computational biologists and molecular biologists and biochemists learn to speak a common language?" And so we're going to have 2 panelists, Brenda Rubenstein and Juan Alfonso. And so hopefully, and obviously we want you to discuss, be involved in the discussion as well. And then after that, to conclude, we'll have a result. So before we begin with the first speaker, just some housekeeping things. The bathrooms on the floor are behind me, so you can go walk here. And for the speakers, you have 20 minutes for your talk, and then we'll have 5 minutes to transition and for a couple of questions. And then hopefully we'll also have time for more questions during coffee breaks. And so does anyone have any questions? No? Okay, great. So we'll start getting ready for Fairbrother, who is both in the RNA Center and also in CCMB, and has been here since 2005, since the inception of CCMB.
Nice. Okay, good. All right. Okay, great. Okay, so I like this quote. I found it last night. It's by Charles Darwin. Let's see. Great beginning. Not that. That's actually the first thing. He was referring, I think, to his fellow scientists, not the species trying to extinction. And so I think that I kind of like that. That's kidding, honestly. Because I think computational biology is a relatively new field, and I came around along with Soren the year after the CCMB had been created, and it was definitely made up of scientists that had a former life doing other types of things, and so there was a lot of adaptation and coming together. Sort of computer scientists, applied maths, and statisticians study kind of problems related to biology. And it was sort of driven by, I think, as Lily said, the Human Genome Initiative, because just a huge amount of data sort of flooded the field. So it made possible the sort of like analytical techniques that data scientists use. So data science is kind of a thing now, but back then people were sort of just piecing together different approaches that had largely been borrowed from other fields. So it was kind of like a nice kind of piece of history for me to witness. So I'm going to talk today, not like a super sciencey talk, but just more about my lab's experience, like in this like space right here. And I'm also going to give a little pitch for the COBRA to talk about support for the COBRA that's available to researchers in the CCMB and to scientists working on RNA biology projects. And so, if I may, just really quickly, this is the COBRA. So this is a small center within the Giuliani RNA Center. I almost said the Rudy Giuliani Center, but it's not that one. It's basically kind of an operations center. It's kind of the organizing principle of the RNA Center, but this is a COBRA-funded grant, an NIH-funded grant to kind of support junior faculty to do research projects in kind of intersection between RNA and other types of fields. And it's also a core where we do sequencing, and we offer services to the community, and everybody here is welcome to use those services. I put this slide up because a member of the CCMB gang was kind of one of the original authors of each one of these 5 junior faculty gave a small research project that got bundled into this application. And kind of the mixture of sort of computational data science approaches with traditional RNA science approaches is very attractive to the kind of like reviewers, and it's kind of a winning formula. So I hope we can kind of grow in this direction in terms of new faculty hires and so on. So I just wanted to say that. Then I'm going to sort of talk about this like divergence of different projects that started with me coming around in 2005. I did my postdoc in Chris Burge's lab at MIT. Did a lot of like computational methods for characterizing cis elements, particularly splicing of cancers, using different camera analyses and approaches. Chris was mostly known for GenSpan, which is this HHM model for identifying genes in the genome. I got into doing variant analysis. And we try to use different population genetics techniques to ascertain selection on different types of variants. The first project I'm going to just briefly go over is this work I did in collaboration with Ben Raphael, who was a member of the CS department here. We developed this method for positional distribution analysis. And the idea here is you can learn a lot about a cis element by looking at its distribution around annotation. So for exons, for example, you have an exon and 2 introns. Exonic splicing is a auxiliary sequence that helps recruit splicing factors. And it's abundant in introns. So if y-axis here is frequency, we'd see a elevated distribution, perhaps peaking near its life sites. And it's less abundant in introns. So you get a picture like this. We used a Hamer approach where we fragmented sequences into all of its component Hamers. We used hexamers in this particular case. There's 1,096 hexamers. And you can compare the distribution of each hexamer around the 200,000 exons that are in the annotation. So you get this kind of picture, and you can compare one hexamer to another hexamer. You know, they're both ESCs, they might have a similar picture. But if one of them is not in an ESC, it might not really respond to the fact that it's in an axon and have the same sort of distribution. when you can measure the distance between these two vectors, which is nucleotide position and frequency, using some sort of distance metrics like an L1 distance. We then use this to do K-means clustering. We identify different clusters, and you can see when you look at the different clusters, known elements of splicing fell out. And at the time, the benefit of this was you could also predict mutations, sort of leveraging the distribution of the context of the motif, which mutations were disrupted. So, for example, if you did this trick with CAA-GAC, and you wanted to consider a mutation with the A to T mutation of the number 3 position, you could look at these two different distributions. And you can see this is very similar to the kind of ESE distribution. It's enriched with exons relative to introns. It's enriched closer to the splice sites and further away. But the mutation has almost like an opposite effect, right? So the single point mutation, that gamer has a completely different genomic distribution. It's enriched in introns, and it avoids being in the exons close to the splice site. And we find that this is because it acts negatively. So in splicing mutation, if this mutation occurred in an instance of this gamer at this position. So we tested this experimentally, and fortunately it worked. And we have a splicing reporter which has a mixed phenotype where complete splicing is this band. You can see when you put the wild type version, which contains this gamer, into the middle exon there, you get an additional amount of splicing here. However, when you get the mutation, you get simply splicing from the first exon to the third. And so that's a classic phenotype of deleterious mutation. And you can see that it effectively there's mutations. We then went on to apply this method to sort of evolutionary questions. So instead of comparing one word, the distribution profile of one word to another word in the human genome, you could compare the distribution of one word in the zebrafish versus the human. So you can do pairwise species comparisons. And you look, how is this tamer distributed around the exon? And so tamers with a high distance from each other would mean that that tamer has a different function in that genome than the other one. So we tried this experiment, and we found that most of the tamers were actually pretty similar and had low L1 distances. However, there were a few outliers here, and these outliers were these simple dinucleotide repeats that are often used actually as polymorphisms in fish genetics. So when we actually looked at what the distribution looked like, we saw that we had this peak here, so it was clearly an intronic splicing answer for the 5' splice site. When we looked at the GT, it had the same distribution on the other side. So putting this together, we found that they actually base pair with each other. So these sequences are complementary. So about 3% of zebrafish introns have this complementary pair that brings the splice sites together. So it was a use of secondary structure in this other species. We could use the available genomes at the time to see that this was mostly confined to fish, although we did see it in lampreys too. And it was absent in species on either side of this in the tree. We've repeated this since we've found other simple gamers that make the same kind of secondary structure bridging in other species. So we really had an interest in sort of trying to understand individual cis elements and mutations in the structure from a sort of computational standpoint. We also wanted to use experimentation to kind of confirm that. So we developed in 2017 a massively parallel reporter assay, so a massively parallel splicing reporter assay. So this assay basically used an artificial gene with a common first and last exon. And we would use synthetic oligonucleotide synthesis to ligate pools of exons, both the wild type and the mutant version, into the internal space, and then test these in parallel. So kinds of experiments that we did were looking at hereditary disease alleles. So we used the HGMD database, and we engineered 5,000 different hereditary disease alleles that were in exon states of this middle exon. And then we analyzed these mutations either in vivo or in vitro. So in vivo, we simply put a CMV promoter and introduced this into, I think, Pec293 cells, and we tried different cells too. We extract the RNA, and by PCR, we amplify this and we see which exons are included, and we can calculate PSI values, enrichment, wild type versus mutation. Most of the time, the mutation is not really different from the wild type in terms of its inclusion. However, in some cases, there's a gain or splicing. More often, the mutation goes to loss of splicing. So about 10% of the exonic hereditary disease alleles describe splicing. When you do it in vitro, you can actually visualize the spliceosome forming on the substrate. So you can in vitro transcribe this single RNA, incubate it in heliconuclear extract, and you can see the A and the B and the C complex form on this. So this is like worked out biochemistry from like the 80s. And you can see where the mutation blocks spliceosome. So you can get mechanistic insight into how these work. And one of the things we found was there was a great diversity of sites where the mutation acted. So in some cases, mutation acted early. In some cases, it acted late. So here's an example of mutation active early. Here's an example of mutation active late. And these values of allelic ratios of mutant wild type at these different stages of this A, B, and C complex. And they represent the progress from starting material to product. When we clustered these and we looked at mutations that occurred in the same exon, we initially thought that these mutations, different mutations in different cis elements could cause the inhibition at different stages of assembly. But what we found was this pattern is sort of private to the exon. Each exon would fail in the same way, regardless of mutation that disrupted them. And so exon level features were really important in sort of variant prioritization and understanding which variants were going to affect splicing. We had this assay, so we used it. We found did it with cancer mutations. We found that there was an increase in splicing mutations in tumor suppressors. They kind of dominated that with their post-loss of function. Exon skipping with post-loss of function mutation. We also did it with Neanderthals and integras variants. So using an artificial reporter is great for an extinct species. We don't really have the opportunity to do any other thing. We found, unsurprisingly, that splice disrupting mutations were the greater purifying in the human lineage. But we also found in specific stories of integras variants appearing to, like, through alternative splicing, confer benefit to humans.
5 minutes.
Oh my God, I'm serious. Okay, so we kind of kept on going in this direction. We did it with asymptomatic individuals. We got about 1% splicing. 1% of the alleles affect splicing. Basically found there are certain exons we call hotspot exons, particularly susceptible to splicing mutations. So if you analyze them and like splice AI and put in a bunch of different saturation genesis, a larger proportion of mutations will affect splicing according to the prediction. We then kind of went back to annotation. Branch points were completely unannotated. We developed a method with Lariat-seq which exploited strange circular reads that you find in RNA-seq databases. So when the first transcription reads through the sequence, you would get an inverted alignment, and the point of discontinuity is the branch point. So you can use RNA-seq reads to actually identify where branch points are, and we can start to map branch points in human genes. We worked with a group at Rockefeller, where it turns out that if you have a mutation in the machinery that degrades lariats, so you get excess lariats, it creates an issue, susceptibility to viral infection, where like influenza and HSV1 can be lethal diseases. We just recently published this work showing that that was because of endogenous double-stranded RNA caused by inverted repeat alums that are in about 25% of all introns. So this flood the cytoplasm, they attenuate the PKR sensors. And it just, I didn't realize I was so short on time, but like, with a method like Lariat-seq, you're not going to annotate all the branch points in the genome. So we started to look at ways that would work complete. So we're fortunate enough to collaborate with Dr. Sling's lab, who's on with the PhD lab, created this deep learning ensemble for branch point prediction. This is kind of the architecture of the model. This is the kind of validation scheme where we validated branch points of 80,000 neutrons, which didn't have experimental data. You can see that about 2/3 of the validated instances in the MPRA coincide with the predictions of this deep learning. And also see that in an artificial system where we randomize the branch point in a particular intron, so we randomize the branch point and look at all possible sequence variants, there's a correlation between the model's predicted use of the branch point and the actual experimentally determined use. So the higher probability the model gave it that actually more efficient in splicing. So we tried to combine this kind of machine learning with actual experimentation. One of our goals is to create annotation for branch points. So this is the USC genome browser. We have the annotation with the computational predicted experimentally determined branch points. You can see this good overlap. We also, as part of the annotation of the mouse over, we look at mutations and get a sense of how much mutation will disrupt the solvability of the branch points being used. This is output of the model. So I've kind of talked a little bit about lariats and looking at the species. I've talked a lot about analyzing mutations computationally with Taylor analysis, with high throughput functional assays, and then also talked about sort of this novel algorithm for identifying these rare species in rare but obligate species in splicing. And I just want to briefly talk a little bit about the order of intron removal, because this is a work done by Sissy Han, who was mentored herself and also Soraya Estrella, and she won the another name, the CCMB prize winner. I don't know if it was a wand or, but like she did a great, fantastic job. And it was really building off this 2017 work which she did way better than we did it originally, which used the idea of removing a paired end reads, where you can get two reads that are separated by a variable distance to observe cases where one intron splices before another intron. Right, so if you have this read that spans a splice junction and this read spans an unspliced intron, you can count that as one observation for intron 2 being spliced before intron 3, which is a blue circle. And it can happen in reverse here. And what Sissy found, what we found earlier, is the splicing doesn't happen in first come, first served way. Is often in a particular order or a bias towards a particular order, where you can go through these intermediate states and we hope to continue this work, trying to understand what, what are the kind of orders and what makes for dependencies between intron processing events. Okay, so this is sort of the last slide. This is an example. So remember, red means downstream intron splices before the upstream intron. So you can see that plus gene is a very strange gene that really starts splicing at the 3' end of the gene. Seems to splice, kind of retro splice. There's a lot of questions that come out of that. With that, I'll thank the people in the lab, Luke, Jack, Brady, people in the program, Marcelo, Scott, Ceci, obviously, also Erica for working with the Cobra, Soren for helping to mentor Ceci, Brian, David, other graduates who worked this year. Showery, postdocs, and you did some of the stuff. Former lab members that did the projects who I covered. CCMB faculty, Bass and Federica for helping to support you, and Ben for working with Jared for his contribution. David, Sabine for working with Stephen and Amelia. Collaborated throughout the universities places. This is kind of the distribution of splicing orders with adjacent intron pairs, where about 20% of the events are all within 100% they splice first or they splice last in relationships. And so this is so this works being supported by the NIH and so on. So I think I don't like to take questions because I think yes.
One question.
So, super fascinating, really intron ordering work. Do you understand the molecular mechanism of sort of what's actually happening?
For the order of intron removal?
Yeah.
So that's a great question. It's very it's a very new field. Experimentally, we have established some cases where an intron requires another intron to process. So if you made constructs that would sort of mimic the potential intermediates, so if you had one of these introns that are always spliced last, and you just put it by itself in sort of spliced version, and you put it into cells and see if it splices, it doesn't splice. So there's actually like some sort of dependency. It could be complexes made from the earliest splicing where you get deposited on the RNA and then can function, but the history is required because we put the sequence there and it doesn't tell. So there's also ideas about kinetics. Is there sometimes just one spliceosome on the molecule, because that would also be potential for a kind of extreme event like that. But like we're really trying to figure that out.
The splicing is around those would be incredible.
Sorry,
if there was if there was wrapping back, within the splices of a complex, like that would be fascinating. We should be able to see that in the splices and evolution.
Just wrapping back.
It's also like the
reverse
introns are sort of ordering within the splicing zone itself. So like they're all connected.
Yeah, there may be. There may be. I mean, generally, you can get one splicing event. You can make these chimeric transcripts, and they kind of recapitulate the original things. I don't think there's this elaborate design where you have something a sequence here that tells this part what to do. Because if you chop these up, they mostly it's something else. I don't know.
The splicing order, you know, you repeat the questions.
Amelia asked, is the splicing order concerned across species? I wouldn't think so, because there's so much sequence divergence in the introns, including intron sizes. Different species have different mobile elements and repetitive elements that can really influence stuff like this. So I would think no. But there may be particular cases where I think it would be very interesting if we did find genes that was concerned, because that would be a real strong argument on gene or something that you could, that might be a good way to get it as a problem. Yeah. Oh,
You're able to visualize spliceosome formation in lifetime? Is that?
There are different ways. What we do is we use the time stamping approach. So we put like a base modifying enzyme onto a spliceosomal protein that has, that's part of a particular complex. And then you can kind of measure the dwell time of that complex by looking at the degree of modification. So you can use the degree of modification as a proxy for time of contact. So you can kind of piece together, that's not really, there are, Melissa Moore had these biophysics things, single molecule stuff where that are quite artificial, but you can actually see the individual events with different fluorophores and stuff like that. So there are possibilities to do that too. But they're kind of restricted. You can't really do it in vivo on natural substrates.
Right, let's take now Bjorn Sanstede was airfly man, isn't it?
So thank you very much for coming to the talk. This is work that my group has been doing over the last 3 or 4 years. I should say that I'm an applied mathematician, so my endeavors into computational biology only started really 5 to 6 years ago, very slowly. So I'm going to talk about 3 different projects. The first project with Erika Wenchen Zhao, who was a postdoc at Brown, is now at Wake Forest, and she really did the majority of the work. She did 99% of the work. The Tambara thing in CS here, and then Victoria Antonetti is a student of mine who's now following up on this work. And then 2 more projects that are really important to touch on. Stanley and Yen are also PhD students in my lab, and they're working on 2 different projects that I would really just like to brief outline what they're doing. So that's the plan. So what we really do is, as I said, I'm an applied mathematician. We are really trying to develop methods, in this case for understanding single-cell RNA data. So we want to design algorithms, for instance, to align single-cell data across modalities. So you want to have data from chromatin accessibility, single-cell RNA data. You want to, in this case, actually, you have, as I said, you have 2 axes that you can use to measure both. But if you can't, you want to align these data and predict what the different measurements would have been if you have been able to measure both at the same time. And then more generally, infer meaning from single cell data. So the way that we try to do this is to build algorithms that have mathematical foundation. So we really want to understand not just construct an algorithm and see whether it works. We want to understand why it works, when it works, also ideally when it does not work. And that's kind of aspirational. Sometimes you have a mathematical foundation, sometimes you can say very specific predictions about when these algorithms may or may not work, but often we really have to start thinking about sort of indirect indicators that tell us when an outcome, the result that we might get, might actually be true and when it might be false. And so the example that I'm going to focus on today, one of these algorithms asks for mathematical foundations to be transformed, and I'm going to talk a little bit about this in the context of the first and the second project. So the first one is in inferring gene variable networks and single cell data that are timestamped. So we have sample information about when these data were collected. Then the second topic is cell-fate prediction during cell differentiation. And then the last one is understanding spatial gene transcript differentiation, in this case in decimation data. So that's the plan. So the first one is thinking about DNA networks and how we can infer them from single cell data. So you might imagine that you have a process, you have cells that you have sampled at different time points. There's different genes that up or down regulate other genes as the process evolves. You can get gene expression counts again, timestamp bins. And what you would like to do is go from these expression counts back and for which cells up and down regulate which other genes. So it's a very simplistic picture, but the idea is getting so. And so the way in which we're trying to do this is we're using gene velocity. So there's many different algorithms available that people have used. So our approach was to use gene velocities, the rates of change, to make this inference. So the idea is, so the hypothesis is that you have two different genes at two different timestamps. One of them might go up at the timestamp 1, the other one might go up at a later timestamp. Then the hypothesis is that means might mean that gene A upregulates gene B, and then if you reverse all these values make it different, you can have top-down regulations at different points. So this is kind of the idea behind this, that if you can understand gene velocities by itself, we might be able to infer something about how which genes up or down regulate. So what that means is we don't really have any weights. We have data connected different timestamps, so we don't really have any velocity information. So the idea is to estimate gene velocities, understand how genes might change, how gene expression counts might change from timestamp to timestamp, and then use that to predict these regulations. And so what I would like to do is just walk you through the algorithm that we developed to do that. So the first thing is, in order to understand gene velocities, we need to understand gene cell trajectories. So we need to understand how cells would evolve in time if we were able to measure them at distinct time points, which we can't. So if I have like a cell population at one time point, a different cell population at different time points, and I would sort of draw them apart, so I just sort of indicate that they're different. And in this case, this would live in so we basic collections of gene expressions across all genes. So this would live in some high dimension space. If I have 5 genes, this would live in 5,000 dimension space. So I just put 2 of those here in the HPV. And what I would like to do is I would like to predict where these cells might lie relative to these other cells at a different time point if I had been able to measure them with data. So for this, I have to understand like how I could map those. So what we're trying to do is predict cell trajectories at a data statement. So maybe what we might get is something like this. Say it's over here. So these 2 close to these 5 close to these 4. And so the principles that we use to do this is there's really 2 of them. We want to minimize the changes in gene expression vectors. So I don't want to take a cell here and move it down here unless I really have to. And I want to move nearby cells to nearby cells. So I think about cells with similar gene expression vectors should evolve in similar ways. So I don't want to take those 2 and move them apart. Right, so these are the 2 principles, right? So just like again, what we don't want is I don't want to move this one down here and this one up unless there's something else that forces that to do. And I also don't want to break up or destroy geometry. So if cells are nearby, initially they should map to nearby cells. So optimal transport is a way to accomplish this. So more specifically, this what's used for classified optimal transport. There's 2 different cost functions in there. So we penalize making drastic changes to gene expression vectors. Then you also penalize destroying the geometry. Those 2 you can put together. And then you can look at within this framework, minimizing these 2 cost functions, you can basically get a probabilistic map from the cell population at 1 to the next time. I'm going to show you the formulas for this if you're interested in a bit. But the idea really is like the way to write this down and then calculate those maps. So it also allows, this entire approach allows for unbalanced cell numbers. So the number of cells can be balanced from one to the other to understand. And yes, you don't get necessarily a one-to-one mapping. You get a probabilistic map that maps essentially distributions of cells at one time point. So once this is done, we can basically take a cell at one time point. We know where you predict the cell to be at the next time points. So now we have this trajectory. Now we can the velocities just by finding them. So we just be a rate of change. So we have that. And then we can basically do use that to estimate a gene interaction matrix. So we want to predict from the gene expressions at one time point for a specific cell how gene expressions at different time steps for a different cell might look like. And we want to average this out essentially to get a regression in a regularized way of all the different cells that we have, so that we just get something that builds from genes to genes. So a map that maps genes to genes and tells us how each gene at one time point impacts the gene expressions of the data. We're doing it in such a way that we predict velocities, not gene expression. And once you have this, you get a matrix. The sign of the entries in the matrix tell you whether the prediction is up or down regulation. And the size of the absolute value is in some sense an indication for how strong that regulation is. And the nice thing about optimal translation is that probabilistic expectation that goes through this process here. So let's think about this as a significant level. So how significant, how confident are we in that prediction? So now thresholding the absolute values, so taking only the largest absolute values gives us measures that we are more confident in. And if we take all of the possible entities into account, we get a more complicated network, but we have less trust in it. So in this case, here, one, which is the first column, for the up-regulating to the down-regulating. So we can come up with class. So that's essentially the idea. And so here's just sort of the formula. I'm not going to go through this, but here's sort of the formula how this works on the optimization process where we have two different cost functions. These are the cost function animation. This is minimizing changes, this is minimizing distances. There's a parameter in there that says how much to value one over the other one. That's something that we have to figure out how to set this. Then from this probabilistic mapping, we can actually map cells to new cells. So we take our second projection. The gene velocity estimation is exactly what I said before. It's to find a difference. And then we have two different ways. I just talked about it in the regression, but also do kind of correlation. Two different ways to predict these matrices. So they're actually, and they have both have their advantages and disadvantages. The second one is very easy because it is summing up terms. That's very fast. It's also very scalable. The first one is the minimization. That's very expensive, but it also has a lot of advantages. It allows us to minimize the number of edges we predict. And so that typically gives you for smaller networks much better predictions. So that's the trade. So maybe just two slides with like comparison with our algorithms, and then one with what I'm going to do with these kind of methods. So these are two different data sets, data sets that are based on biological networks, but are basically generated. The data are generated using Boolean networks. And so what we can do is we can apply existing algorithms, which are sort of HTTP here, and then use our algorithm depending on the two different inference methods that we can get use and compare them. So what I'm showing here is the methods for the two different data sets. And then this is the area under the position depot curve. So this is essentially what we're calculating there is if we know that there is an edge in there, what is the probability that we actually correct that and we get that, and then we get all of the actual edges. So how much of the B quality we get? What is the number of edges that we know are there that we actually count? And so ideally, this way is usually as high as possible. There's a dashed line in there that's random, a random guess. So these algorithms, all of these algorithms don't do actually very well. If you think about overall value, right? So if you just flip a coin, it will be here. Right? So that's the way it's the same, like you're not much better than panelists. So you want to be as far away as you can. But you also see that you're not that far away, right? So you get a base rate twice. Random guess, so it's a typical scenario. So algorithms are inherently just very complicated. There's just to be large number of genes, large number of edges. It's kind of hard to get. Okay, very accurate. But you see that our tool in 31 compared to the second path that is already not together. So this is something that is just one scenario we show using a data set that comes out of Federica, questions now to Sophia. Yeah, we try to sort of use this for just checking that algorithm gives you something that's confident about. And I'm just going through on this lower part here. So you can look for each gene, you can see how many edges go into it, so how many other genes would regulate it, or I guess would predict they're regulated, and how many other genes that gene would be predicted to be regulated by it. The ratio basically tells you the ratio going to a point edges when you're in the graph. If this is small, you think that this gene is an upstream gene. If it's large, then it's an downstream gene. And here's just like 4 different genes for which the predictions are coming first for generally, but these are 4 genes for which the location upstream of the ocean is known. And I want to predict that that's it. So that was the first project. So the idea really was using a way to predict gene regulation using single cell RNA data, and doing this in a way that we have an algorithm that is not a black box, but where we know exactly each step what it's doing, where we can assess validity and where the algorithm might break down. And I didn't talk much about how we do this, but that's sort of been the optimal transport frame. So the next project is it's really in its very early stages. So I'm just going to talk briefly about what we're trying to accomplish in like this one single result that we have so far. So this is predicting cell fate from single cell data where you have an site initiation. So this is just a sketch, right? So imagine you have some precursor cells, they already have decided to some extent which cell type they want to become, and then at a later point during the development process, they basically achieve. And so you can think about this as being PCA or EMA projection. What you would like to do is, without having information about when these measurements were taken in the development process, you want to identify that there is a differentiation that's happening. You also like ideally to predict for each of these precursor cells which of the cells they will be. So you want to predict the cell fate. And so in this, a number of different algorithms available, most of them gives information about the times at which these cells exist, or which part of the development process they are in. So we don't want to do this. So the way we're trying to think about this is we're doing hypothesis testing. So this looks like a tree, but that's precisely what it this way. It could be maybe more complicated, maybe it's just a effect, maybe there's no differentiation going on. So ideally we want to test different models for what might go on with these cells and then decide which of those. I'm just going to not do this. I'm going to decide on one single latent space, one single model. In this case, that's a tree, because that's what you think should happen here. So I'm going to pick a model. In this case, it's a tree, one single branch point where your cells differentiate. That's my latent space. What I would like to do is I would like to match those cells to the latent space. So I want to map them onto that latent space in such a way that ideally we want to separate cell types into the different cell types in the process. And so the way we do this is again on Wasserstein transport. So we can compute distances here. I can take any 2 expression vectors and compute like how far or different they are. I can also along the tree, I can compute distances as well. Just how long this take the other one. And so that's exactly the setup that we used before. So I can just map the cells onto the space. And I'm plotting them here. These are not, they should all lie on that tree, but then if I collapse, see anything. And so the hope is then that if you're doing this, it preserves that tree structure. In this case, that's in the data. And so it should separate cell types. So that basically gives us a tree structure. It does not tell us which of these cells would go up along which branches if we're able to follow that every time. So what we're going to do is we're going to now distinguish time. So we're saying that I'm thinking about this being time, let's say 0, this is time equal to 1/2, the 2 endpoints are time equal to 1. What I'm trying to do is use this as a pseudo time on my latent space. So maybe this is time 1/2 of the 4, like a time axis along this that splits into these 2 elements here. And now I'm going to do the same thing that we did before. I'm going to map this onto the tree, but at the same time, for each of these points here, so in each of my data points, I have now a pseudo time associated, and I would like to represent these pseudo times correctly. And I would like to take this branch here and move it into 2 different parts, one for the cell type, for the first cell type, the other. Instead of looking at those together, I would like to separate them and force my cells to decide on which branch do they lie. So I'm just going to do it like this. So I go from 0 to 1 half to 1 for my first cell type, from 0 to 1 half to 1 second cell type. So again, I have pseudo time here, same as before, but now I'm forcing these cells here to decide where they are. And then I do the same thing. So it's now exactly the same algorithm that we used before. It's two different cost functions. One cost function is for the difference between what to preserve the geometry here, and then the other one is what to preserve the pseudo time. And so then that would just hopefully enforce the cell themselves, and that would tell us where they are. So basically, the idea is that choose a latent tree structure, latent space that you want to test for, and then introduce the pseudo time, long living time, and then map both in a way that basically tries to explain both of them. So here's one data set where we use this by Weinberg. It's a differentiation of white blood cells between neutrophils and monocytes. So what they did in the experiment, they had complete information about the cell differentiation process because they used a lot of clone cells, so they knew exactly they could trace them through the differentiation process. So the colors here are basically colored by the total fate bias that comes from the experimental setup. And we can basically, forgetting about which cells are which, we can basically use the same algorithm. So the first step would create something like this. We see that predominantly that monocytes here with these two exceptions, and then they mixed here from the undifferentiated cell population. And then if we're now going through this process, then actually we separate. So we have basically almost there's outliers, but that step works very well and separates out cells. And we can map those back to here and see where they are, which I'm not going to show you, but you really see that the more towards the end you are, the more towards the end you are here, and the population usually becomes from the undifferentiated cell population. So in this, here we actually have it one tree, so we can see how well we're doing. So there was this one parameter alpha that decides whether pseudotime or the cell structure preservation is more important. So over a pretty large range of alphas, we get pretty much comparable results. That's good because it's quite robust. We don't need to worry about how we're choosing this. If we're emphasizing pseudotime too much or the cell geometry too much, then it doesn't work. That's what we know. And then we get essentially like close to 70% accuracy, which is better than they have in the paper. And so again, this is a more realistic approach. We're not going to talk about this, but you can also set probability boundaries based on this one. So I think I'm running out of time. So, just one slide very briefly. That's the last project that the end has been working on. This is trying to understand spatial gene transcript localization, where specific genes might have the transcripts localized within the cell towards a specific region. Maybe the transcripts lie more closely to the nucleus. In this case, we want to test whether they are more close to the sinusoid blood vessels that basically separate like between these cells. And the data that we're using is actually human fluorescence data. So in this case, it's the paper by Novoselsky or people by Novoselsky. They basically use learning to identify cell boundaries and to annotate bins that lie close to the or on the sinusoidal blood vessels. What we would like to do is we'd like to test for specific genes whether they're meaningfully localized closer to the sinusoid or not. And so the way that we're doing this is, the end is doing this, is assigning for each bin that we have, assigning the distance to the next sinusoid, to the closest sinusoid as a provider, so as a spatial coordinates, and doing the same thing for the nucleus. So we can test whether there's nucleus distance or distance to the sinusoid is more relevant for predicting where majority of the transcripts lie. Then basically using so distribution that's parameterized by the different providers. It's distance to nucleus, distance to sinusoid, and then fix that. And then the idea is that looking at the fit allows us to test which of these two providers, which combination of providers explains data best. And in this case, what she found is that if you're looking at over and under expression at these kind of coordinates distance to nucleus distance. So you see that distance to the nucleus doesn't really make any difference. And everything that is the real part that makes a difference is the sinusoid. And you see that, sorry, the closer you are to the sinusoid, the more overexpressed you are. So that basically indicates that in that specific gene, talking about will be expressed or will have skills much closer to the sinusoid than elsewhere. So you can do this for all possible genes, and then get but we found that the huge advantage of doing this with the way that is doing this, that's fitting them in is it could be a gene that is expressed strongly here, and you would see this in this data. If you just average this out, you would not see anything. So it gives you very specific information in terms of its components for what the localization might look like. So that's with this, I'm not going to go through this. I think the main challenges for us really are validation. How do you make sure that what you got is correct? And ideally, you want to do this internally. You might be able to have some branch data sets where you can test, but what we need is based on within the method that we can calculate and indicate how confident we are in the results. And that's something that
That's very interesting. The question, I know this is kind of for your analysis, but the velocity measurements. So those are sort of inputs you use for no right? Do you have issues with certain genes don't really take good velocity measurements and what it's do? Yeah, and it's kind of proving that useful in some of the reading you try to do it like does the post processing.
Yeah, so the question is like maybe there are some genes where velocities are more easy to calculate or have a more clearer signal, while for other genes maybe there is no clear velocity prediction. In principle, we can do this, and we can look at the variation of the prediction of the velocity across all of the different cells. Take a gene, look at the variability. We haven't done that, but we could definitely do that. So the other way, which I talked about this, but another way, you have RNA velocity available, and that will be another input that either replaces or supplements the way in which we estimate G velocity. And that's something that Emporio is working on, whether combining that with RNA velocity will make a short algorithm that's more accurate or but in principle, we have information from the estimation part, and we could do it. We just have some. So we can. Right. So in this right. So the method that they basically use is they basically have a binary classifier for each bit. So you either here or you not here, and then you can make it basically do a hypothesis test on estrogen distribution. Is that a classifier or not? The problem is that you the signals might be very low, at least very few transcripts per bit. Your classifier might be wrong. So there's lots of different points where just the algorithm might break down and not give you the right answer. And so this is in some sense it's more accurate because it cares about how close you are, but it doesn't care about like assigning a binary classifier. And it gives you more information about. So for instance, imagine that maybe this is a gene where it's doing those things. Then on average it doesn't do anything. But this method would detect the other one. So you really get more detailed information about how the different covariates, that's the case, like two different distances affect or are related or correlated with the localization activities. So in principle, what you can also do when this is interesting is so in these two because in this context that's what the test would have. But you could also look at first different distances and then put them as covariates and see what the outcome is. So you can really do a much more certain that does not rely on classification a priori, but really where you can just Okay, so let's think of more possible ways. Ideally, maybe identifying these biological axes from the data and then testing how the station is related to that. That's it.
So, the floor is yours. Thank you so much, Emilia, for organizing this. My lab works on pathophysiological mechanisms using animal models of disease. And I'm going to use some of our cardiac data to give you biological context and highlight the areas for which our work will be strengthened by collaboration with the computational group. So I'll go over each of these areas during my talk. But again, to give you a little bit of context, using the heart as a system, we're interested in understanding how this organ responds to stress. And there are various phases of remodeling that can occur, for example, in condition of increased workload, such as hypertension, or because of a genetic background that is pathogenic, or perhaps a metabolic disorder, and so on. So there are these mechanisms that lead to remodeling and eventually heart failure. And we're specifically interested in how cardiomyocytes, which are the contractile cells of the heart, are responding to stress. And we can recapitulate remodeling at the level of cardiomyocyte by looking at how the cells change in shape and size. But it is also important to keep into account that when we work in a system like an organ, and in this case the heart, when we are interested in cardiomyocytes, in the sample that we use, we have a heterogeneous mix of signals coming from different types of cells. And all of these different cells, and I'm just mentioning here like the most abundant, like inflammatory cells, fibroblasts, and endothelial cells, are all responding to stress and get activated and participate in remodeling. So with this in mind, even independent from the cell type you're interested in, it is clear that gene expression is altered during disease. And we are interested in post-transcriptional mechanisms that can explain gene expression changes and help us to understand what determines the lack of linearity between mRNA abundance and protein levels in cells. Now, in this regard, RNA binding proteins are specifically important regulators of these processes. And now you can imagine that if you have RNA modifications, these may change the affinity of RNA binding proteins for these transcripts and therefore affect the transcript fate. Now, in this regard, we need to keep into account that there are close to 200 known RNA modifications of which we know very little about. And for the purpose of this talk, I'm going to tell you a little bit about M6A. This is a well-established modification which is correlated with disease in various systems. So I'm only going to show you here in the context of the heart how the level of this modification is increased in dilated cardiomyopathy patients that underwent an explant, so a transplant for a new heart, compared to donor human hearts. Now, we have used, just to give a little bit of a background, so this modification occurs internally on mRNA and through an enzyme called methyltransferase 3. This is a reversible reaction. And once the RNA is modified, it can be recognized by M6A binding proteins, which can then control various steps of the life of mRNAs. So to understand the role of this modification in the heart, we have engineered the mouse models to have either loss of metal 3 in cardiomyocytes or higher levels of this pathway. And what we have shown is that these alterations are sufficient to perturb homeostasis in the heart. So using metal 3 in cardiomyocyte drove a dilated cardiomyopathy, so heart failure. And increasing metal 3 also changed the size of the heart, which is not good when you think about it. It can also be a risk factor for heart failure. So in this regard, we've shown that this pathway is important. And to understand what mRNAs are M6A modified in the heart in a metal 3 dependent way, we had a couple of years ago collaborated with a group in Germany, Dietrich and Heidelberg, who perform nanopore sequencing, which is a method that allow us to not only directly sequence RNA, but also gain information on some modifications, including M6A. So what we found using these methods is that within the transcriptome, the modification can be found throughout the body of the mRNA, with preferential sites close to the stop codon and the 3' DR. What was also interesting is that the modification was not randomly distributed in the transcriptome, but it was enriched in transcript that encoded for regulators of mitochondrial metabolism. So in this regard, we asked the question, is then this pathway important for metabolic stress responses in the heart? And I'm just going to show you very few data on this, but we use Western diet feeding of the mice as a metabolic stress. So this is a high-fat, high carbohydrate diet. And what we found that is interesting is that, well, there was a smaller global reduction of M6A in general in the heart after this stress, but also the location, you can see here in red, it changes. So you use a little bit of that enrichment, for example, at the stop codon. Now, we've subjected mice that are deficient for metabolic, specifically cardiomyocytes, to a Western diet. And what we found is that we saw a systemic phenotype. This is very unusual. So we saw that this change in cardiomyocytes had an impact in obesity onset. Here in gray, you can see the eye line is the percentage of body weight increase in control mice subjected to Western diet. And this response was blunted with cardiomyocyte-specific metal 3 deletion. So this is not common for the heart to be able to have the systemic impact. But overall, this suggested that metal 3 had an impact on both local and systemic metabolism. And again, so now you have a scenario where you have a molecular change within cardiomyocytes. You are affecting an epitranscriptomic marker, which can obviously have an impact on the proteome. We have a disruption of mitochondrial function, which therefore will also induce metabolic changes. And altogether, either metabolites or factors secreted by the heart are also communicated in distal organ, overall creating an interorgan crosstalk. And I'm going to use this information to go over some of the initial, perhaps bioinformatics challenges that a group like mine that is not computational is facing. So the first one is, again, having the ability to integrate multi-omics would be important, because you cannot just think about looking at the transcriptome itself, will not be sufficient to understand proteomic changes. But in this case, we also have pathways that affect metabolites. So again, thinking about metabolomics, proteomics, and transcriptomic data in the context of these epitranscriptomic changes would be important. And again, this I'm using the heart as an example, but you can think of every organ system, any syndrome will also involve the interaction between different organs. So while we can think about the integration of different omics within a specific tissue, we also need to think about integrating data from different organs. Now, there is another information here that could be valuable for us is that, so while single cell data is feasible in certain systems, for example, single cell analysis of cardiomyocyte is not possible. These cells are very large, we cannot flow sort them. So the only information available on these cells is from single nuclei. Now you can imagine that if mRNA abundance is already not a good predictor of protein levels, now if you're only looking at mRNA levels in the nucleus, then you're even farther away from a physiological relevance of the data. But there is a ton of data overall out there that can tell us what signals may be deriving from cardiomyocytes versus fibroblasts versus endothelial cells or inflammatory cells. And I believe if we have a way to deconvolute the data using bulk data, but then use available data sets to at least gain some information that help us predict what are the changes that could come or are more likely to come from cardiomyocytes versus other cell types, it will be very important. And obviously, there are also other issues that for transcriptomics, there are single cell data, but that's not the case for other modalities at this point also. Finally, okay, we are looking at RNA modification, for example, F6A. But again, so we want to understand the processes that this modification are regulated. So having a way to have data from the modification, but also be able to correlate it with an outcome, so a process, an RNA stability assay or translation. So again, be able to really get from the modification to the mechanisms. I mentioned that this modification is followed by methyltransferase 3. This is a predominantly nuclear event. But when mRNAs are F6A modified, their fate is controlled in the cytoplasm by RNA binding proteins that can recognize this marker. And the YTHDF protein is particularly important for this. There are 3 family members, YTHDF 1, 2, and 3. And this has been shown to really have a unique consequence when they are engaged in a transcript. YTHDF2 drives degradation. YTHDF1 has been shown correlate with I guess, pro-translation. YTHDF3 have been a more elusive function. But with this, for example, one of the strategies we can use, okay, we can again engineer mouse model, knock out each of these proteins. And in this case, we did proteomic analysis to see how the proteome is shifted uniquely when we lose either YTHDF1, 2, or 3, specifically in cardiomyocytes. So we can use this data. We can also correlate this data with cardiac functional data, showing in this case that we have a cardiac phenotype when we lose YTHDF1 and 2, and not so much when we lose YTHDF3. But this is just an example of something that we're doing. What we can also do is looking at, okay, what mRNAs are bound by YTHDF1, 2, or 3, using pull downs and sequencing the mRNAs that are engaged by each of these proteins. By doing that, we can understand if there are shared transcripts. This is just an example of some of the transcripts that are bound by each of these proteins. There's some others that we identified. And we can also see unique signatures of the transcriptomes that are bound by these different proteins. But this is a very simplistic approach. Again, the truth is that when we think about orchestration of F6A modified mRNAs, we need to think about our time and space of this regulation, because we believe, for example, that these different proteins have a unique function and effects on mRNAs because they are also located in different subcellular compartments within the cytoplasm. So we can see this with immunostaining. So having a way to gain information that is also spatial and temporal when it comes to RNA binding proteins and engaging with their mRNAs will be very important. And right now we're just looking at one protein at a time in one time space in a bulk way. So again, it could be related to RNA stability or translation. Is there a localized effect out of each of these proteins, how it is changing with stress during stress conditions? And there is not really a unified model that can link all of this information together. Now, moving on, I mentioned obviously some of the data on mRNAs, but I also want to point out that to really understand the regulation of the proteins and how it can be modeled after stress, we need to keep into account regulation at the level of translation. And now in this case, non-coding RNAs, such as transfer RNAs, are central. And these transfer RNAs are amino acid native but amino acid RNA synthetases. And adding the right amino acid on the right tRNAs is essential for proper decoding of the genetic information. Now, because I mentioned RNA modifications, we need to take into account that mRNAs are neither the most abundant or most modified molecules, and transfer RNAs are highly modified, and we still have little information on these. So we're also interested in this case in understanding tRNA dynamics. And I believe there will be a talk on this. I don't need to go into these details, but there are unique bioinformatic challenges when you think about studying tRNAs and understanding perhaps cell-specific pools that are available, the stoichiometry of RNA modification, the charging ratios, and how all of these biological effects can play a role in stress responses, also in a complex system such as the heart. And eventually, just to segue, I'm running out of time, but obviously we need to also understand translation and connect these to, for example, how perhaps cell-specific tRNA pools are also controlling codon usage in specific again, cell-specific way, so connecting regulation of translation. And for example, a group like mine, even when you think about ribosome sequencing, we just don't have the expertise to really get to this type of analysis. And now I want to use this information to just have one last slide, which I think is very important, which really gets to the connection between or the potential connection between silent variants and pathogenicity, because currently all the gene-wide association studies are really focusing on variants that are changing the corresponding amino acid and are predicted to be pathogenic because of that. But we could argue that a variance that is not changing the amino acid, but is changing the codon that the cells has to use, may have an impact, perhaps because of rare tRNAs available in the cell. Or in this regard, also, you may also have an impact because you may change now the sequence for an RNA modifying enzymes, and therefore by changing that, you can have a strong impact on an mRNA, for example, even if you cannot predict it by just looking at the amino acid change. So in this regard, if we have a way to have a framework that can go from synonymous variants to codon changes, tissue-specific RNA availability, predicted functional consequences, it would be, I think, very innovative and clinically relevant. So altogether, what I wanted to get at is that the challenges that we are facing go all the way from tissue level integration and RNA modification mechanisms, all the way to really the genetic interpretation of clinical data that are available, but we don't understand fully yet. I'm done. I just want to maybe mention that all the tRNA work that we plan to do currently, for this we are collaborating with Alfonso, and Jess specifically will give a talk on this. And in Nanopore, we had a collaborator with the University. More recently, we are using the RNA Cobra with Fairbrother and Jack, and they are helping us with that.
Like additional proteins that you're looking at your system, the IGF-2 EP proteins.
Marginally, yeah, we're very interested in that. We don't have any more models for it yet, but again, yeah.
Well, I guess one.
So it's yes. Okay, yeah. So the first question was related to other proteins that regulate M6A. If you're looking into those. And the second question relates to what would we need to, or I guess, what could we do to predict the silent variant as a function of consequence? And yeah, again, it's a little bit tricky, but there are examples. For example, a study shown that a silent variant on a gene did not change the amino acid, but it changed M6A formation. And that, in that case, it had an impact on mRNA stability. I think based on clinical data, we would have to just do correlations first, and then we need to go into maybe a mouse model and then really look at functional relationship. But the data, there's a ton of data in terms of like genomes that have been sequenced, and only a very small percentage of congenital heart disease can be attributed to the variants that as a consequence in terms of changing an amino acid. Everything else has been disregarded so far. Yeah.
All right. It's also part of the RNA Center, and you learn the RNA Center. And you
Yeah, so my group is shy of 1 year. We started in July last year. In my group, as you can see from this first slide, studies control protein synthesis. That thing down there on here, that's actually polysomes, so ribosomes and mRNA making a protein that we isolated like a couple of years ago by Jesse, Eric. But in general, my group is actually interested in any step that regulates the process of basically making the RNA, basically degrading the RNA, and creating proteins of the RNA. And you can see this is a current and some former members that contributed to these studies. I should point out that this is a collaborative group now, like for my wife, that is also in RNA Center. And we also do quite a lot of collaboration with other groups that are interested in ribosome structure in the translation in very weird organisms. But I would say my majority of my group is in these three projects which are looking at the proteins that regulate gene expression that basically contribute to changes in translation, initiation, and elongations of protein synthesis, as well as how they are controlled by other like RNA binding proteins, so the short RNA. The central topic, as you mentioned, is then RNA. So this is the scheme of eukaryotic RNA with all the things that are associated with it, and also how the translation of protein synthesis occurs. One thing that you'll notice there are these UTR or untranslated regions, and these are actually where majority of the gene regulation occurs. This is essentially where the microRNAs, all these other non-coding RNAs bind, as well as RNA binding proteins, and they regulate some of these processes, either initiation, elongation, much less termination. So when I started my group, I focused on microRNAs, and the point was essentially to look at the mechanism. So they really went biochemically into the mechanisms of microRNA, and we show that this process occurs like this. But as you can imagine, like the cells actually don't have only microRNAs, there are a bunch of RNA binding proteins, and essentially in 3' UTRs, it's always maybe a competition or like synergy of these molecules working together. So you can imagine that this is a very, very kind of like modulating process, or I would say like process that you cannot really decompose easy. And for this reason, I think they've already introduced the event from a single reporter, is like just a single mRNA, to a multi parallel reporter assays, which actually give us an insight, basically like what this can be. And you actually have multiple kind of like regulatory elements in one mRNA. But the whole point of these multi-parallel reporter assays is actually to make models, like mathematical models, that basically we know what we are putting in, we are barcoding these reporters. We can put them in the cell, we can get multiple things like we can get translational efficiency, this polysome fraction of like mRNA, we can get translation initiation from this like 4DS subunit like association, or we can get generally like RNA stability from the like just count things. And the reason for this is basically to get like good kind of like controlled assays so that we can actually move it and then analyze things globally. So the outputs of these things indeed shows similar things in different cells, as you can see here. This is a familiar regulation of these reporter assays. This is a microRNA regulation, and you can notice there is a difference between different cells, and we can immediately put this like globally with the different cells because we know that microRNA expression, for example, for this particular microRNA LED7 is like essentially lost in neurons or neuroblastoma cells, which are these M2A cells, but present in all the other cells. And then you can see a very complex kind of like distribution of like where these reporters like fall into the RNA expression levels in the different cells if they are regulated by a originals. And this is essentially like reason for this is there are 11 different A-rich binding proteins in like cells, in cells, and they are not expressed in every cell, and they are not expressed equally in every cell. So it brings complexity of like how you need to look through these data. And this is actually something that's even further kind of like by accident, by sequencing accident, we found out that A-rich elements are not only controlling the stability of the RNA, but they are actually controlling also splicing, something also Bill talked about. And what you can see here is essentially the presence of these A-rich elements in this kind of reporter would activate cryptic splicing of this reporter, which basically is purely dependent on these A-rich elements. Now, you can say this is an artifact of the system that we created by just making these reporters, but Courtney, that was doing this before from Baylor College of Medicine and Tony Muslim group, actually found that this same thing exists in human cells. And what she found is essentially that depending on the rich element that's found here between these two like exons, is essentially like they'll create this REC1B isoform of this protein, which doesn't influence the REC1 protein to begin with. But if you delete a one protein that regulates these rich elements, you basically lose completely REC1B. Why is this important? Is because this process of REC1B is actually controlling this epithelium to mesenchymal transition, which is basically a hallmark of cancer. So it's not only REC1B, but she found like essentially like thousands of things like this coming from the multiple reporter assays, and just looking globally how this looks like. So obviously, we use this data like to like look at the other things, and these are some of the things that we have open questions about that are again heavily computational based as well as experimentally. But you can imagine we can do now like NPRA essays or global studies in 900 different RDPs that we know. We can look how the mutations in these things are actually influencing like diseases. We focused originally on the neurodegeneration, autism, and cancer, and then we can intervene. And this is something that I want to put forward. And this is collaborative project that we started looking how we can actually rescue insufficient mutations in the patients that would have like gene gene diseases coming from it. The idea was essentially if we know good prediction of how the microRNAs and RBPs are binding and where they are binding with nuclear, we can block them. And essentially, because majority of these things are down-regulating the protein production and gene expression, if you're actually blocking the microRNA or RBP or both of them together, you could actually alleviate and increase the expression of a gene from a single allele. So this is something that we want to do in a gene-specific way, so targeting one gene and not all the others. And we want to do it in a cell-specific way. It's obviously missing now. But an example of that is basically this TDK1, that is actually a gene that is mutated heavily in ALS. And we basically started by just random targeting nuclear with ASOs, but we also have computational studies to predict RNA binding protein sites, microRNA binding sites. Then we actually find the best possible region to target with this short antisense oligos, and you can see we can actually come up with a ASOs that upregulate 1.5-fold or approximately 3-fold on the protein level. And then we can move this to patient cells. We can move this to essentially like fibroblasts that we get from the patients, and you can see that if you are getting a fibroblast from the patient, it has like reduction in a TDK1 mRNA or in the protein, we can actually alleviate that expression by just introducing this antisense oligo. It's not only for ALS, we actually got contacted by multiple groups. This is the most four recent things. We were targeting post-dating. Post-dating is interesting because of muscle wasting, especially with the GLP-1 inhibitors now. So we can actually create an ASO that will upregulate muscle production in the mouse in a matter of 4 days. Or we can actually target BRCA1 by mutations and upregulate BRCA1 in the cancer patients. But there are a bunch of other things, and Majid, who's here in audience, is doing majority of these ASO studies. Now, this brings me to another point where we are actually looking at the motives in mRNA, and we are looking how they regulate translation. And majority of these projects are actually focused on this strange thing that these structures of A-rich nucleotides can regulate elongation, and they are regulating it actually by dampening the protein production. So what we found over the years, and the multiple people involved in this projects, is that, as I said, runs of phase in the coding sequence of these human genes would downregulate essentially production of the protein or also mRNA stability. And they figured out the mechanisms of this, and one particular mechanism that we were interested in, like here, is that essentially, besides splicing, you actually can get multiple proteins from a single gene by frame shifting. So you can actually get one gene from one gene, one mRNA, 2 different proteins, and they can actually have 2 different functions. So this goes also under the control of stress. So we notice that actually heat or any other stressors can impact these sequences, and they actually are there to basically reduce the effects of the stress. So Jeep, John, and Sam are looking at these things in different cells now and with different genes. One example is collaboration in Amber-Stratman, where we noticed that this gene that has such sequence basically regulates splicing of cilia and centrosome genes, and loss of this genes basically reduces expression of cilia in zebrafish, and you can see the phenotypes during the development of this zebrafish. But interestingly, we could rescue all these phenotypes by just keep stressing the fish. So actually, if you grow fish from 32 degrees, which is a constant fever, essentially, you would basically rescue not only the phenotype, but you also rescue splicing or splicing cilia. Now we push this even forward to the CK, and we notice that actually loss of this gene is actually reducing splicing, and we are creating all sorts of problems in splicing. One of them is this huge kind of increase in the circular RNAs that are byproduct of splicing. And by heating, you can actually downregulate it, you bring it back, arguing that like splicing is somehow regulated by them. So we also need computational studies for this. So we are collaborating a little bit with Bill, with a couple of other groups on this, like looking at how these genes are regulated, what are the introns that are spliced out or not spliced out during these studies. And now we have a data set of like both zebrafish and human cells where we are looking at different temperatures. So this is happening in absence or presence of this protein. Now, again, there are a bunch of different projects here. It's not only time, but actually we are actually introduced the synonymous mutations and complement deficiency as a problem for genetic disorders and cancer. And then we are really interested in this part here, how these genes contribute to regulation of the stress. The last but not the least, over the last year, we published this study where we basically went from like this tedious job of actually getting like things from a dates, and not only one day, but actually it can be a week, to isolate ribosomes and ribosome protecting fragments that are needed for ribosome profiling to a single R. And this is mainly work from Jesse and Danielle, that essentially did this while we were at WashU, but now Nicholas and Sam are looking like to help Jesse and others to actually extrapolate this work even further. What I'm saying by this, we are basically using just the magnetic beads, polylysing magnetic beads to isolate all the RNA and ribosomes, translating ribosomes from the cells. And this is beneficial because now you can actually get structure like this one from Cryptococus neurformans that Jesse did like last year. In 60 minutes, you basically can go from a sample to like basically cryo-enter it. And you can get the structure of a like ribosome from like a very scarce sample. This is just one structure that we published. We have like around 15 to 20 structures that probably that we still didn't publish that are coming from a different species. There will be like very interesting structures from mosaic ribosomes and other things from collaborators. But one thing that involves more besides structures and where we would need computational kind of like thing for the like solving structures, cryo structures, is a problem with ribosome profiling that Fed actually like introduced. This problem is basically mainly because you need really high amount of the cells because of loss of material that you are doing during your practice. Essentially, you need to start between 10 to 30 million cells, and you lose essentially 95% of that by just doing this sucrose pushing, which we don't get in our like prep now. So we created a new method. We call it RAPL-seq, not RIBO-seq anymore. And it's basically just using these magnetic polylysing beads to basically scoop up everything from the lysate. We do digestion either in the lysate or on the beads. And then you basically like do normal ribosome profiling step. We optimize it currently. I would say this is pushing the boundaries. Like we would do repetitions with 100,000 cells regularly, but we are going further down because there are some steps being in. This is kind of like from the landscape in preparation, and you can see again like this like difference. Basically, if you go to the sucrose gradient, you are losing 84% of your material just by actually loading your ribosomes. And the sucrose gradient, we can actually rescue around 98% of them by double elution using our things. And even if you want to intervene here with the RAPL, after the gradient, you would actually rescue 90% of it, because essentially like you lose additional steps during the precipitation. And this can be seen here if you're using 40 million versus 500,000 cells, and actually quarter of that loaded in the gel, we get more fragments here than from 40 million. This gives you a nice distribution of ribosome protected breeds, which actually allows you to look into a translation of every single gene in like these cells. And this is the periodicity that we get. This is from 500,000 cells. You can also see that majority of these things are basically associated with translating or mRNA coding sequences, and the reproducibility is almost 99%. This gives us the possibility now not only to do tech cells that I'm showing here, but it's essentially like we go to a biopsy of 1 to 5 milligrams of like tissues. This one is here mouse brain, essentially. We did it also for mouse lung, and you can see a specificity that essentially only neuronal genes are expressed and common genes, but these ones, as you can see in scale, are basically barely present. We also did it in the brain, in the lungs, so you can see that these genes come up while neuronal comes down. And this is not a single cell thing, because here we are covering all the genes, all the transcripts, not the ones that are basically just highly expressed like you are looking at the signals. The other point that I want to make, we don't really need to go after the just tissues. We can actually go after the tumor and adjacent tissue. This is the resection of the tumor from the patient, where we can actually compare translation in the normal tissue that is surrounded versus the tumor. And again, we need 1 to 2 milligrams of the cell or the tissue that is liquid nitrogen frozen. We are pushing this now to blood samples. So we are collecting blood samples under with Juan Alfonso and Bennett Garnero and Matt Hartfield, basically looking at the melanoma cancer patients, to see how we can follow essentially protein synthesis under the treatment or without the treatment or during the metastasis. And I would finish there and basically just give shout out to all the people that did and a bunch of collaborators that we are working with improving these methods for these projects and also funding if we receive them. That's how they think.
So the bead method, is that mostly polysomes you're getting?
So the last question with the bead method is polysomes that we are getting. So no, we are getting actually all RNAs. So what I didn't have time to talk about. We do mild YZ, so we are not getting nuclear fraction. We can get nuclear fraction if we want. So in this fraction, we are getting tRNAs, we are getting ribosomes and mRNAs and everything that is inside the protein. So we have projects where we are looking at sequence tRNAs, peptidyl tRNAs, also the ribosomal RNA. The ribosome protected fragments are coming after RNase 1 digestion. So essentially you would basically deplete everything that is not protected by ribosome. And that's actually pretty good because then you can actually even use it either in the lysate or the beads. Yeah.
I have a question about the heat disrupting the frame shift mutation. I was wondering about this earlier, frame shift, whether you think the RNAs are being sort of stress granules to because it's not, it's a different loss function.
Yeah, so I mean, it's a valid question. So I will repeat the question. Is it the heat and frame shifted associated with the release of the things from the stress granules or being kind of then translated or not mRNAs from stress granules? So no, we can do it in vitro and in vivo. So what we noticed over the years is basically that once you actually heat the ribosomes, they start actually making mistakes more and more. And what we noticed is basically that heat stress is basically, especially these monotonic sequences like poly-A or A-rich sequences, would actually create even more problems for ribosomes. And in some cases, not in all cases, these new products are actually rescuing your cell. So actually they are preventing further damage to the cell. And that's what we basically are studying. So essentially how these frame shift new species are actually contributing to cell response to heat stress.
Thank you so much. Hi, everyone. Next. So this session, we have two trainees speaking. And the first one is you, who's a CCMB PhD student who works with Yale Law. And she's going to tell us a little bit about her research today.
Thanks for the introduction. So I'm Bjorn. Yeah, right.
So you might want to speak up just.
Okay, I will speak louder. Yes, okay. So today I will introduce one of my recent research project to develop a model with complexity and structure in called Novel-ST, a deep learning framework for automated label transfer and novel cell type discovery in the spatial transcriptomics. So let's start with a little bit of background. Spatial transcriptomics recently gained some popularity in the community because it enabled the measurement of gene expression while preserving spatial organization within the tissues. So among various technologies, we can divide them into two major categories. One of them are sequencing-based technologies, where the tissue uptakes and the assay and we can form the next generation sequencing at each spot. Therefore, these technologies can combine high gene coverage with limited resolutions at spot level. So the alternative will be imaging-based technologies, where we attach the fluorescent probes to the RNA and use the microscope to image and detect the RNAs. So here are several examples of imaging-based technologies, including Merck's and Cosmic SMI. So one key advantage of such technologies is that it can achieve at a single cell resolution. It means that we will get a gene by cell matrices of gene expression along with their spatial locations. And this enables the reconstruction of the spatial gene expression patterns. So given such data sets, the next crucial step will be the cell type annotations. Meaning we assign each cell a biology identity that based on its gene expression profiles and some known markers. And so this will be also an important intermediate step for lots of downstream analysis. And I include several examples here, which are differentiated expression gene analysis, co-expression analysis, cell type specific spatial variable gene detection, and cell communication inference. And there are lots of computational cell type annotation methods. And among those methods, reference-based one recently getting more popular because it can automatically transfer the cell type labels from a reference data set to a new target data set. Specifically, we expect the labeled reference data sets have well-curated annotation labels that coming from the marker genes and the expertise knowledges, so we can directly transfer the label to a target without any human manipulations. However, these methods have an underlying assumption that all the cell type present in a reference should also present in the target. Actually, I say it reversely. Like all the cell type present in the target should also present in the reference. But in real applications, this underlying assumption is not always guaranteed. That the target tissue may contain some normal cells or normal cell types, which have cell type that are absent from the reference data set. When this happens, the reference-based method will to assign these normal cells into an existing cell types, rather than recognizing their true biological identities. Therefore, our method are focused on overcoming these limitations. We can develop Novel-ST, novel cell type discovery and annotations in spatial transcriptomics method. Specifically, Novel-ST taken the annotated spatial transcriptomics data sets as a reference and want to annotate an unannotated spatial transcriptomics data set. It simultaneously transfer the labels from the reference to the target and identify the normal cell types if they are present. And there are some existing methods which are also working on the same problem. However, they share some common limitations. So firstly, all of them require some user-defined parameters as input on the model. Specifically, spatial ID
depends on a manu-Trojan threshold on the classification probability. And Stella also requires users to input a parameter that specifies the expected number of novel cell types. And Accornero also further requires both the true number of cell types, which including the existing number of cell types and the novel cell types. Therefore, in their applications, users will encounter issues about how to specify that specific hyperparameter. Additionally, all of them doesn't have the uncertainty quantification to assess how reliable the allocation label we got. Therefore, our method wants to resolve both challenges. And here is a motivating example to show why these applications are quite important. So Stella is the top performing method on the cell type identification. And on this example data, we wrote Stella for multiple runs by specifying different number of novel seed, which is the expected number of novel cell type. It turns out the results vary a lot, even different number of novel seed. Specifically, when we specify that number to wrong, which is the wrong truth of number of novel cell type, it identifies very minimal number of novel cells and achieving a very low F1 score. Alternatively, when we increase that number to a higher number, such as 5 or 9, the F1 score increases by identifying the majority of the novel cells. However, it starts to misclassify one truth of novel cell type to multiple of them. Therefore, it motivates us to develop a method that doesn't require any user-specified parameters that can automatically correctly identify the true number of cells. And then here is the pipeline of our method. So it's a three-stage pipeline, where the first stage we learn a latent space, where we hope the reference and target data set are well integrated there. And the cell types form as cohesive clusters. And during the second stage, we transfer the labels from the reference to the target data set. And during the third stage, we calculate something called confidence score to identify the low confidence cells and revert them to the normal cells. And this is a motivating example showing we can correctly identify the selected group of cells, which is a ground truth of the normal cells. And I will discuss about each individual stage. But during the first stage, we use an autoencoder to learn the latent space. And then firstly, we have a reconstruction class, which is trained on the reconstructed cell expression. And additionally, we want to add constraints to enforce the cluster structure in the latent space. So specifically, if two cells belong to the same cell type, we will enforce the intercluster apartments. Specifically, we will minimize two cells' cosine similarity and the Euclid distance in this space. If two cells coming from different cell types, we will enforce the intercluster separation by encouraging the Euclidean distance to be greater than a margin. This is a final loss term, which composed by three components. The first one is reconstruction. The second and third one are meta consistency loss and min-max feature loss. Each of them enforce or minimize the cosine similarity and the Euclidean distance respectively. And we have ablation study to show that all of three loss terms are playing effects. So specifically, when we remove one of the loss terms at a time, we show that they have a higher DBI score compared to the role model. Specifically, this metric measures the cluster quality. So if they have strong intercluster compactness and the intercluster separation, it shows lower values. It's showing that our model actually optimal when we have the all loss terms. And during the second stage, we transfer the label from the reference to target using a graph-based label propagation algorithm in the latent space. And specifically, it's underlined in the assumption that if cells are closely with each other in the latent space, they should share the same cell. And the third stage is the most important stage of the model. Specifically, we computed confidence score to identify the normal cell type. The confidence score is composed by two components. The first component is central proximity. Since we found that normal cells are tended to lie far from all the centroids of existing cell because they are not belong to animals. So we compute the distance from each target cell to the centroid of existing reference cell types and transform that score to be higher if it is closer to the existing cell. And the second component, we measure the neighborhood homogeneity. Is we found that if a cell is located between two at the boundary of two clusters, it's more likely to be a normal cell type. So to capture this, we calculate this. So the final confidence score is combined of these scores, and then we apply a Gaussian vector model to cluster on the confidence score and select a cluster with a minimal mean to be the normal cell type. In addition, we also consider there are cases that there are multiple normal cell type exist, so we perform a subclustering with a scale model. And this summarizes our method section. And then we designed a comprehensive benchmarking pipeline to evaluate NOVA-ST's performance. So during the quantitative section, we apply a pseudo-normal setting, which will be the setting in the core control. Specifically, I mean that I remove more of multiple cell type for the reference to be the true normal cell types. To select the cell type to be removed, it's only two criteria. So first one is cell type proportion that will remove the most abundant and least abundant cells from the reference. And secondly, we also found that if cell type is similar to existing clusters or different from existing clusters, it also changes the cluster difficulties. So we use more separated and also mixed settings. So we evaluate in total of 4 settings. And then we benchmark our method against 5 existing methods. Specifically, single RNA is a single cell RNA annotation method. Telegram is a ligand-based label transfer method. Spatial ID is a deep learning-based method. And Stella and SO are novel cell identification method we mentioned by Mencoff. And I evaluated cell type annotation performance with 4 metrics, which are adjusted rate index, accuracy, weighted F1, and macro F1. So on this example data set, which is a false Merkers primary motor cortex data set, which will know as T consistently outperform the existing method on all 4 settings. In addition, we also evaluated the novel cell type identification performance by itself, which is a binary measurement between novel versus non-novel cell types. So we use a total of 7 metrics, and the results are showing in this breakdown. So for this benchmarking study, we in total evaluated 9 data sets across 3 platforms, 3 species, and 6 tissues, where the detailed information are showing in this table. We found that NOVA-ST consistently outperformed the baseline methods across all the data sets. And in addition, NOVA-ST can also accurately recover the spatial. Specifically, in this most primary motor cortex data set, we found the confidence score showing a low confidence as this L2 and L3 layers. And it turns out that this represents the inhibitory neurons as its regions. And NOVA-ST accurately annotates these novel cells, and given its annotation, recovers the unique differential expression gene as this region, while given other method annotation, we cannot do that. So this shows that NOVA-ST's capability to recover the biological information. So going back to this previous example, we show that NOVA-ST can correctly identify with one novel cell type and achieve a reasonable F1 score. So this highlights Novosti's novelty where without relying on the user-defined parameters, we can achieve good performance. And in addition to the quantitative analysis, we also apply Novosti on more realistic data sets. Specifically, we're interested in if we're using a healthy data set as reference and transfer the label from the healthy to the disease data, whether we can recover the disease-related cell types. So we evaluated in total of 2 data sets, and I'm showing one example here. So for here, we're using a healthy mouse collar as reference and label the inflamed mouse collars data set. Given the ground truth of subtype information, we can see that there is a cell type shift from the immune cells from reference to the target data set. Specifically, the target immune cells show high proportion of inflation-associated immune cells, which are the neutrophils, macrophage, CXL, positive, and monocytes. So we transfer the label from reference to a target, and then we analyze so given our labels, we start to analyze biological identity of the annotated novel cells. So firstly, we perform the differentially expression analysis, and it reveals that so the EEGs in this novel one pathways are mostly represent the marker genes for those inflammation-associated cell type, including neutrophils and the macrophages. And secondly, the ground truth of subtype labels also further confirms this information. Specifically, the majority of novel cells are inflammation-associated immune cells. Additionally, we also check the spatial organization of identifying novel cell type. So in the original paper, it identified different niches across time points or different status. Specifically, normal cells are found to be enriched in this new 8 niche, which is an inflammation-associated multiple region. And it have 2 characteristics. One is infiltration of immune cells associated with inflammation. And we can see that most of the normal cells were identified exhibit in this new 8 region. And also, it also showed a big loss of epithelial cells. And our annotation results exactly matches this characteristic. It shows that Novosti showing accurate annotation and showing accurate spatial organization. So this concludes our method where Novosti is a fully automated self-annotation framework which without requiring any user-defined parameters and also quantified uncertainty. And we show that Novosti outperform 5 baseline methods across 9 data sets. And also Novosti can accurately identify the disease-related cell types. So the manuscript is currently under restriction, and here is the link for the GitHub page. So anyone interested to try the method of this guidance. So I will thank Dr. Ma and Dr. Singh for their supervision and also all the members from both labs for their support. And thank you. Okay, so the question is, our method can measure the uncertainty and how large the uncertainty is in our data, in our results. So I would say the uncertainty is depends on quality of the data. So especially if anyone work with physical spectrometry data, we know that there are certain cells which may show low quality because of the progress of sequencing and also when we kill the cells, whether it has small appearance or not. So specifically, if we check the result, we can see the cells which have also high uncertainty usually exhibit within the cluster very clearly, and the cells show no uncertainty are usually on the boundary or spread out without randomness. And this is a way to kind of compare, like the uncertainty is correctly in all cases. But we don't have a very specific value to questions. Okay. Yes, and also, I thought so.
I'm Jesse. I'm a postdoc in one of them, so it's lab of Bjorn. I just recently got data back from collaboration from Federica this morning, so I won't be talking about that project, like she had mentioned. But instead, I'll be talking about something that I worked on during my PhD, where I used tRNA sequencing to map modifications across Arcan. So, like you may have heard already, beyond the canonical bases of RNA, there are RNA modifications. And depending on which domain of life that you're looking in, there's a unique variation to these modifications. Altogether, they help to contribute different functional potentials to different classes of RNA. And the most densely and diversely modified class of RNA is the tRNA. And in this case, they're actually clustered around 3 different functional regions, most famously in the anticodon loop, where they contribute to coding fidelity and codon recognition. And then the modifications in the core help to stabilize the structure and maintain the overall L shape. And then the ones that occur in the T and the D loop help to maintain the tertiary interactions, which are at the elbow of that L-shaped structure. Something that I'll also mention here is that most of these modifications are introduced by a specific enzyme at precise locations. So this is something to keep in mind when we're doing comparisons across several different tRNAs and several different species, and even domain-wide, that they're often conserved at very particular positions, and you won't really find them outside of those areas. So archaea make for a great system to study modifications because they grow at a lot of different extremes and live in a really challenging environment. So for this project that I'm talking about today, I sampled 9 different species, which range from thermoacidophiles, which grow at temperatures around 75 degrees Celsius and at very low pHs from 2 to 3. And then hyperthermophiles, which are comfortable at near boiling temperatures around 95 degrees Celsius. Methanogens, which have a broader range of growth temperatures, but live in anaerobic environments. And then halophiles, which are moderate in terms of their growth temperature, but prefer salt levels that are near saturation. So in order to compare and analyze these modifications from all these different species, I need to use a high throughput approach. So traditionally, mass spec
is the gold standard for mapping modifications. You get an unambiguous signal that you can map back onto the tRNA and get the exact chemical identity of that modification. The only downside is that it's very low throughput, so you can only purify a small number or even an individual tRNA at a time, and then analyze it with mass spec. Alternatively, one of the other methods that you can use is tRNA sequencing. So here I'm only talking about RT-based sequencing. There are other methods like nanopore, but I'm focusing just on reverse transcription. In this case, in a single experiment, you can collect an entire pool of tRNAs in one go. And then instead of getting the exact chemical identity, you're inferring the modification through reverse transcripted signatures like misincorporation. But this gives you site-specific resolution, even though it doesn't give you the exact chemical identity. So something when you're doing sequencing is that you always need to have some sort of reference, either that or using an orthogonal method for validation of those misincorporation signatures. In this case, there's an example, Archaea helophorax volcanii, that has had nearly almost every one of its tRNAs purified and analyzed with mass spec. And so in this case, I'm able to take those misincorporation signatures and map them back to a reference to determine which modifications are detectable. And this shows that if you look at the heat map, the ones that are highlighted with the asterisks are the ones that are known positions for a given tRNA. And the pattern for misincorporation occurs specifically at modifications that disrupt the Watson-Crick base. So those are modifications that will interfere with canonical base pairing during reverse transcription and result in a really strong signal of misincorporation. At the same position, there's some that also interfere with that phase that are not as bulky, and so they'll result in fewer misincorporations or a lower rate of mistakes that are made during reverse transcription. And then you'll notice that a lot of these that are compiled here on this cloverleaf structure are actually invisible to the RT. So that's one of the drawbacks, is that if it doesn't interfere with that face, then it's something that we wouldn't be able to detect using the reverse transcription. So those include pseudouridines, NYC, and a lot of the 2'-level manipulation. So fortunately, many of the species that I sampled had at least partial mappings of their tRNAs using mass spec. So this is a summary of just the number of mods that have previously been mapped. And like the one that I showed you from volcanii, it's far more than all of the others, where there's more than 320 reference positions that you can go off of. But for the others, we can still use the sets that are available and propagate annotations across tRNAs that have not been analyzed with mass spec. So in this example here, I'm showing the initiator methionine from eukaryosis, which has its known set of modifications that are used as a reference and the misincorporation signatures that are associated with them. And so those can then be mapped to a homologous tRNA in a closely related species that has not had its tRNA analyzed. So in this case, these aren't called confidently right away as certain modifications, but they're candidates. So there's a couple of other steps here that I'm not going to go into detail, but I look at the background distribution of misincorporation across all the positions, as well as the intensity of misincorporation to make more confident calls and determine a score for each of the predicted modifications using this homology-based mapping approach. So this is just a summary of the number of annotations that were gained from using this approach across each of the species. So for the ones that had no annotations previously, they now have several hundred.
And then for the ones that were already well characterized, like Accornero, there's fewer gains than were already known, but there is still over 100 that were filled in the gap in terms of what was missing from that reference set. So a bit of validation when comparing the misincorporation signature between these closely related species, you can see that there's strong conservation between all of the tRNAs in those conserved positions. And so most of them are likely the same type of profile when you compare them when you compare two species that are closely related. But then the outliers themselves are really informative in this case because they're candidates for either different modification types or it might be the same modification, but it is present at maybe different levels of abundance. So you do get approximations depending on the modification of the stoichiometry. So I'm going to focus on this example outlier here from the initiator methionine, which is known in P. furiosus as M22G, and is a predicted candidate in Kodak rings. So in zooming in, it's not just the intensity of the incorporation that's higher, but it's also a different pattern of incorporation. You can see that there's more deletions that are occurring at this position than there are in reference. And so one of the candidates modifications that occurs at position 26 is this trimethylguanosine. And what's unique about this is it has an additional methyl group on the trigger, as well as the ones that are interfering with Watson. So to explore whether or not this is a signature that can distinguish these two modifications together, I pulled together all of the known methylated guanosines of both types. And you can see that the pattern of all of the known trimethylguanosines are, if you squint your eyes, kind of similar to the intensity difference that we see from that single example that we were looking at. But then when we break it down, there's actually significant differences in the composition, particularly in the T and the deletion fractions. So this doesn't guarantee that this is what this modification is. It's not telling us this is exactly that trimethylguanosine, but it's at least showing that there's significant difference in their patterns of misincorporations that could potentially be used to distinguish them apart. And then the single example that you saw between those two closely related species is similar to this, this pattern of misincorporation. So while I'm talking about this trimethylguanosine modification, it's actually not something that would be unique if found in Kodak lenses. It's a characteristic mod for all hyperthermophiles. It's been characterized to increase the structural stability of the tRNA and is proposed as an adaptive mechanism for growing at higher temperatures. And in general, when looking at the profile of a hyperthermophile, you can see that they have a diverse toolkit of different modifications that they can be using to grow in those extreme temperatures, especially when comparing to the modification profile of a mesophile like Meroplutus that only grows at 37 degrees Celsius. But something that I want to focus on here is that a lot of these that are in the acceptor stem are conserved in hyperthermophiles, and normally they would be attributed to, because they're conserved in hyperthermophiles, be attributed to growing at higher temperatures. But I wanted to ask the question, are all hyperthermophile specific modifications designed to increase structural stability like they've been proposed? So when I looked across all of the tRNAs, I noticed that the acceptor stem modifications have a unique pattern of being installed at position 6 or 67, and that almost all the tRNAs have one or the other, suggesting that there's this coordination between the two. And then when looking more broadly across all species, this was something that was independent of clade. It's conserved in thermocoxide, but in methanogens, it's only in the species that grows above 80 degrees Celsius. So it's something that's unique to hyperthermophiles. like I had already told you. And then there's a single enzyme that has previously been characterized to install this modification in that methanogen Mirantiae. But when it was biochemically characterized, it was only tested against a single target, cis TCA. But from propagating annotations, I was able to show that it's in nearly half of the tRNA from the set. And then further evidence for conservation is using comparative genomics to look at the distribution of the homologs of this one characterized enzyme, and not just trying to look for similar sequences, but also looking at the domain architecture of all the homologs to ensure we're looking not just at similarity, but enzymes that are orthologous to the one that's been characterized. And from the analysis, we're able to show that there's a correlation, again, with the enzymes that are present in these genomes of species that grow at high temperatures versus the ones that don't. So in this case, there's no homologs found in halobacteria or in the sulfurobacter. So again, asking the question, why would a hyperthermophile coordinate modifications in the acceptor stem? So in general, GC content of hyperthermophiles is higher than a mesophile. You can see that looking across the span of the tRNA molecule. But something that you might be able to appreciate is that the acceptor stems are especially rich in GC. And so they likely have much more rigid stable structures, which makes sense intuitively if they're growing at high temperatures. But something that has been discussed in literature before is that this modification between the dimethylguanosine and the cytosine disrupts or distorts the canonical base pairing between the G and the C, which would push those bases out of plane with one another and maybe induce some flexibility. So that is maybe what I think is happening here. It's not a complete story. There's still more to be explored, but I think that this is maybe something that's a controlled distortion of the acceptor stem. It's an important contact point for both amino-acyl tRNA synthetases and during translocation in the right. Okay, well, I'm wrapping up. So to wrap up, I didn't present a complete story, but I introduced you to a framework that I think is an exciting tool to use and apply more broadly, not just in RPA, where you can use a small set of references and based off of high throughput sequencing, expand the number of annotations and profiles for known modifications, and then combining that with other methods like comparative genomics, be able to understand and study different relationships between the enzymes that write the modifications and the substrates themselves. So I have to thank my former lab, because this is all work that I did when I was at UCSC, and then the collaborators that grew most of those experiments. That's all.
The sequencing data running like the modification, so we're looking for if it changes
So it's all based off of the reference, and then it's all based off of the mass spec. Oh yeah, sorry. The question was how am I determining from the sequencing data that the misincorporation is a modification? Right. So with tRNAs especially, it's different from other classes of RNA because the modifications are conserved at specific positions. So that combined with reference data sets from mass spec analysis that shows that they're at those sites, it's a little unambiguous. You can't say the chemical identity of the modification, but you know that it's happening at that position. And if you look at the distribution of background misincorporation at all other candidate non-modified sites, there's very little background. So you really only get signal at those quantified positions.
So you must, we have to describe
It's ideal, but you could also use other experimental methods for validation where you could knock out like an enzyme that's supposed to write that. And if your signal disappears, then you can infer that that's the modification set.
So what other systems?
The question was what other systems would be good to use this toolkit with, and I think it's almost limitless. Anything that has tRNAs, right? So it depends on the question you're trying to ask. If you're interested in modifications, most model organisms have at least some set of reference that have been analyzed with methods like mass spec that you can propagate further annotations into where tRNAs with, as long as you're able to sequence.
If you try different races, nations versus their cases, it's possible you'll get different spectrum by using
Yeah, that's a really good question. So one of the things that I was talking about, the patterns of misincorporation, that's something that would change using different RTs. So that data that I showed was very specific to the reverse transcriptase that I was using and would change. And also there's a lot of parameters that you can adjust in the reaction that can change the behavior of that reverse transcriptase, even if it's the same one, and can kind of adjust how many different bases it's misincorporated.
So do you actually do the experiment?
It's been, I use an optimized version and then just consistently use that for all of the experiments. But there are labs that have been playing around with being able to distinguish between different mod types that occur at the same position by adjusting those parameters.
So what's your take on for kind of optimizing throughput and getting positional information and getting information? What combination?
Yeah, that's a good question. So if in this case, the data that I was presenting was from an enzyme that's not commercially available. And so in that case, we would just have to use what we have access to. And so then some of the workarounds for getting the most read through and the most misincorporations is, and not everybody agrees with this, but it's to extend the reaction time. And then just using the standard buffers that were optimized from NED